A complete human genome, the entire biological instruction manual for a person, is remarkably small. If you extract all that raw text, it takes up about 3 gigabytes of data. For decades, science viewed this genetic code through the lens of individual, contained physical blueprints. You sequence a single organism, and you get a manageable, specific dataset. As sequencing technology advanced and costs plummeted, labs stopped analyzing single human genomes. They began sequencing millions of people, animals, and pathogens all at once, shifting rapidly into mass production. While an individual biological record is small, mass production compounded the volume. Bioinformatics facilities went from managing gigabytes on hard drives to processing petabytes of information, catching the scientific community off guard. Exponential data growth triggered a hardware crisis. Global laboratories had to rapidly scale their physical infrastructure to keep up with the daily influx of new sequences. Server racks buried IT departments. Storage capacity meters hit their limits and instantly maxed out, flashing red week after week. Yet the biological payload, the DNA itself, was blameless. The A, C, T, and G genetic letters are compact. The storage crisis was a byproduct of the underlying software architecture. Since the 1980s, biologists have stored DNA sequences using 40-year-old legacy file formats known as FASTA and its successor, FASTQ. Look at this comparison. On the left is a structured relational database. On the right is a FASTA file. It lacks relational architecture, binary encoding, or efficient indexing. It is a literal flat text file. Imagine a major hospital trying to manage 1 million patient records by typing them out sequentially on a single, endlessly scrolling Microsoft Notepad document. That is how genomics manages data. Some of the most advanced scientific minds in the world are relying on primitive text documents to manage the most complex biological code on Earth. Because FASTA and FASTQ are text documents masquerading as databases, total system failure at the petabyte scale is a physical certainty. The legacy storage approach is chaotic, but one specific feature makes it far worse. The FASTQ format damages hardware capacity by introducing a compounding error called Phred quality scores. A Phred quality score is a single ASCII character forced into the file to represent the sequencing machine's confidence for every single DNA letter. Watch the scale in this diagram. For every single letter of actual biological data you save, you must store an equal or greater amount of metadata directly alongside it. A compact 3 gigabyte genetic payload expands into gigabytes of confidence scores. This invisible weight of metadata, permanently attached to an outdated text architecture, is what actually broke the servers. At petabyte scale, flat text architectures break down and cannot be salvaged. There is a technical gap between the elegant efficiency of a biological blueprint and the inefficiency of 40-year-old flat file formats. To unlock the potential of the human genome, biology must move past text documents. The scientific field has to migrate toward real, dedicated database architectures to survive. Scientists recognized the limitations of flat files and attempted the most logical next step, moving genomic data into traditional SQL databases. Traditional SQL databases rely entirely on rigid rows and columns. However, biological data is not a uniform series of records. It is a long, continuous sequence with complex, overlapping relationships. When labs forced a 3 billion character string into a rigid SQL grid, simple comparisons began taking hours, causing the whole system to collapse all over again. Biological data is compact, but inefficient legacy text formats created a massive global hardware crisis. To move forward, biology had to stop relying on text documents and start building real database architectures. Thanks for joining Database History. See you in Episode 5. Never use Notepad as a database.